Welcome to A Shot in the Arm podcast. It's day 2, I think, of AIDS 2024 here in Munich, Germany. And what a day it has been already. So we are coming to you midday after the presentation of really some pretty iconic data. I think it's been quite a day in the AIDS response. And joining us with a different hat on than usual is Yvette Raphael. Yvette, hey there.
Hi, Ben. How are you?
I'm good. What's your acronym today?
My acronym is GCAG, Global Community Advisory Group member for the Purpose 1 trial. But also it's day 5 for me.
Yeah, I know. And it's sort of, I don't know what it is, day 100 for me. Jeff was saying yesterday that he felt like he'd been here forever, that he'd never left. But you'll also, those of you who are watching us in glorious Technicolor, will see that I'm wearing a clean shirt. And yes, thank you, the luggage has arrived. Now, we're also joined, I'm really thrilled about this, to be with us is Dr. Moupali Das, who is the Executive Director of HIV Clinical Research at Gilead Sciences. Moupali, welcome to the show.
I'm delighted to be here. Thank you for having me.
You've had quite the morning.
Yes.
So first up, I think it would be really great to just understand a little bit about this new drug, Lenacapavir, because it's first in its class, and its mode of action, its mode of delivery is very different. So could you tell us a bit about it?
Yeah, so Lenacapavir is first in class. It's a multi-stage capsid inhibitor. It works at both the pre-integration step and the post-integration step. So how I like to explain it to people who aren't virologists is HIV is very wily. It invades our cells, uses our own cellular machinery in the nucleus to make copies of itself. Then it leaves the cell and those copies go out and infect other cells in our body. So what Lenacapavir does is it attacks the capsid, and the capsid is this beautiful geometric structure that HIV encases itself in. It's part of the HIV virus. And Lenacapavir stops capsid from working well. And it works at multiple stages that capsid is required to get into the cell, get into the nucleus, copy itself, and get out of the nucleus and out of the cell. So that's why we say it's a multi-stage inhibitor. And we say it's first in class because no other drug has ever focused on capsid before. They focused on integrase or chain termination or other mechanisms.
Yeah, and given the sort of the life cycle of HIV, we've always known there have been this various points where we could stop it in its track, and the sort of the capsid, the container that it sits in, if you like, has all, I guess, holy grails. And here we are. Now, the drug is already used in the United States. It has approval for use in people already on HIV treatment with extensive treatment history. But the trials today were looking at it in terms of prevention, and our huge doctor, Dr. LGB, Linda Gale Becker from the Desmond Tutu Foundation, she did the presentation. Jeff reported back that there was a standing ovation afterwards. So could you tell us a bit about your research of the compound in prevention and just the top line on what the trial results told us?
Yeah, I'd be delighted to. So is globally for combination with other treatment drugs. And this is really for the small group of people, but significant unmet need in a group of people who have accumulated a lot of drug resistance and don't have other options for therapy. In HIV prevention research, there so far has not ever been a drug that is effective for treatment that hasn't also worked for prevention. So we were very hopeful that lenacapavir would work for HIV prevention, but we were flabbergasted by the top line results, which were there were no HIV infections at all on the 2,000 plus women who were given lenacapavir. So in terms of the research, that's really the main result. This study evaluated 2 different experimental drugs for PrEP, lenacapavir and FTAF, or M-tricytidine tenofovir alafenamide, also known as Descovy. And the results were that lenacapavir was 100% superior to background HIV incidence and 100% superior to Truvada or FTDF, which was the active internal control. And with respect to FTAF efficacy, overall, in all participants, the incidence on FTAF was not different than background HIV incidence, nor was it different than the Truvada incidence. And what that means is the incidence of people who are assigned to take FTAF, who are assigned to take FTDF, that was the same as the incidence that would have been expected in people not on PrEP. So that is overall disappointing, as Linda Gale mentioned. However, if you look at the people, and this was a pre-specified analysis, who were adherent to, who actually took their pills, FTAF was highly protective against HIV acquisition. So we did what's called a matched case control analysis. And we looked at the people who got HIV and people who were very similar to them in geography and voice score. And we looked at what the differences were. And basically the difference was adherence.
Adherence, hey, Yvette. I mean, and that goes right to the heart of the Choice Manifesto, the idea that we have a range of interventions that work for particularly girls and women at different stages. What Moupali said about it's all about adherence, that cuts right to the heart of it, doesn't it?
Yes, it definitely does. But I think for me, what is important with the results today is also is for us to also recognize the role that community played in this trial. And the standing ovation goes back to the fact that women want choices. Women also want their children not to tell the same story. I know Moupali knows this every day. Lillian, Yvette, Margaret, Dazon, all of us tell the Gilead team that this is one of the things that we're so excited about. But the proof of all of this will come to it becoming available in our countries. And I know Gilead has a clear plan on how they're going to do that. But importantly is that it is hope for us. It's hope that our young people will not be infected. So I also want to just congratulate Gilead on this and say we are proud to have been working with Gilead on this road.
If we could pause a bit and your engagement with Gilead in the R&D process, and we'll come on to access in a little bit, but it has not always been the case that women have been able to participate in clinical trials for HIV, and it's not always been the case that African women have been participants in clinical trials, or where they have been, obviously the products haven't been available to them. Yvette, how did the community advisory group support Moupali and her colleagues?
I think one of the first things that we need to remember is that GPP is part of the work that we do. It's important that we have greater participatory practice.
What does GPP stand for?
Greater participatory practices. Yes?
Good participatory practice.
Yeah, but
to be
greater.
for
Good.
You guys, greater. Right?
But yes, but good for organizations like Moupali. But I think for us that was important that we include the community, and these relationships starts at a rocky start. We insist that things happen, but for us this engagement was important because we saw how Moupali's team and the Gilead team listened to some of the things that we wanted to see. We wanted to see women who are pregnant not only get access way after things are approved, but we asked for them to be part of the trial. And we actually worked together on a what did we call that? When they reconsent after,
Yes.
yes, if they fall pregnant, become pregnant, they could reconsent and stay in the trial. We advocated for young people to be part of the trials, and those things were listened to. And it's important that we talk about those things. And our communities, women have always been part of trials. However, this specific cohort of women, pregnant and breastfeeding women, as well as young people, have always been left behind and always only gotten access way after things are approved. But it was a great journey.
So I'm intrigued about the comment of involving pregnant women, because from both a medical ethics perspective and a regulatory perspective, that is always a challenge, a particular challenge. You don't want to put the woman or the unborn baby at risk. It's sort of like a double risk. But
yes, it's our voices, and I think the team listened to us. It's that who is saying we don't want to be participating in these trials. It should be a decision that we make. Who says our babies are more important than any other thing that's going around? We wanted to make those choices, and that's why the reconsent is that women would say, I am pregnant, but what I'm doing right now is for the benefit of myself as a woman and also for my child to see what the difference is. So for us, it was about women making the decision and the ethical issues. Moupali knows women were asking, so do men not die? So why are you worried about us dying because we are on trial? So all of those ethical
I
jargons.
asked the question because it's difficult in the context of established clinical trials. So how did you do this?
I think there's been a sea change in the last 3 to 5 years with the phases program. And I was just on a panel right now where WHO announced the inclusion of pregnant women in antiretroviral research toolkit. And that toolkit is available on the WHO website. And I encourage everybody who wants to include women and pregnant women and lactating women in clinical trials to go review the toolkit. So it required changing hearts and minds in the company, amongst regulators, amongst ethics boards. And I think Anne Lyerly, who's an ethicist at UNC, who really pioneered the Phases Project with people like Dazan and other members of our GCAG, likes to say we should change the expectation from making a case for including women into making a case for excluding pregnant women. And we should protect pregnant women through research rather than from research. And in the case of a long-acting medication, you can't discontinue from study drug, as people in clinical trials like to say. The product, if the person was assigned Lenacapavir and everybody was blinded, so we didn't know, right? But if somebody who was assigned Lenacapavir received their injection and then subsequently got pregnant, there's no way to discontinue it. It's already in their system. So why not follow them within the rigors of the clinical trial environment with all the resources that we have in the clinical trial setting to make sure that mom and fetus are safe? And Yvette and her comrades on the GCAG were with us every step of the way. I first met Yvette in Kigali, Rwanda in December of 2019, before we'd even written the protocol. So we truly followed GPP. And the reason we said good participatory practice is that it's akin to good clinical practice and good laboratory practice and all those other rules that we follow in clinical trials. And that means talking to the people who are most affected, nothing about people without the people.
Right.
And so we met, and they reviewed the protocol, they reviewed the informed consent. There was a pregnant woman who sold sex, who was an adolescent, who was on Truvada. So when I asked the question to the group, should we include pregnant people in this trial, she stood up and gave an impassioned plea and said, yes, because you have to protect people like us. And one way to change hearts and minds is with data.
Right.
And we had excellent data on FTF, on FTDF for years within treatment, within HPV. And we had all of the preclinical data that are listed in the WHO toolkit to make people feel comfortable. So that's, laboratory data and data in animal species, which all companies have to do before they bring something first in human. And we had specific development data, juvenile development, pregnancy, embryology data to allow us to do that kind of work. But what really helps change hearts and minds is stories like the one that I was told, and that I use that story internally to convince people and externally to convince people this is the right thing to do. And people want that. The people we want in the trial want to be in the trial.
Yeah. It's personal for me because going back a few years, I was involved in the in a large clinical trial of women of reproductive age. But should they become suddenly reproductive, in inverted commas, should they suddenly become pregnant, they were automatically excluded from the trial. Like, well, what is the point of this? And in that case, it was an oral medication. It was a pill. You mentioned, Moupali, that that Lenacapavir is not is long acting.
Yes.
And one of the other fascinating things about it is both its mode of delivery, but also the frequency of delivery.
Yeah. Actually, when you asked me about the mechanism of action, I forgot to mention that. So thank you for asking that question. So lenacapavir is a twice yearly medication. It's delivered subcutaneously. So it's not intramuscular. It's subcutaneous, like other subcutaneous products people might be familiar with, might be like insulin is a subcutaneous injection. And the reason we can give it twice a year is that it's really potent. And when I was talking about those mechanisms of action, it's very potent, especially in the pre-integration step, which may be relevant for HIV prevention. We know things post-integration work because integrase inhibitors work for PrEP, but it's very exciting that it's strongest in the early step before the cell gets infected. So that is very exciting. And we've also shared data that shows that the clinical pharmacology or the exposure of lenacapavir is similar in different locations. So in this study, we gave the injections abdominally, but the PK or len exposure is the same abdominally, in the upper arm, and in the upper buttocks. And so when women were pregnant in this study, if they were thin and had taut bellies, they were able to get the injections in their thigh or upper arm or elsewhere. So there were people who did get thigh injections who were pregnant in the study. And we continue to study the alternative sites of injection.
See, that's really important from an access perspective because not a million miles away, there is at least one long-acting, and that's intramuscular, and it's not without its pain. And for listeners and viewers who want to know more about that, you should check out our podcasts with Tommy Williams, who was both a clinical trialist and an ongoing patient with that. So we're almost looking at, and I'm sorry to put it to both of you in such a crass, simplistic way, but it's almost like a twice yearly vaccine, isn't it?
Yes, and we are used to boosters, right? With COVID, people are getting this, took the COVID vaccine and gotten it again. But I want to bring us back to the African's perspective. Why I'm here is that what people see as only a 2-yearly drug, and it's not subcutaneous, right?
Subcutaneous.
It's subcutaneous and all of that. For me and my community, it means I only have 2 clinic visits a year. It means that I get that injection twice a year and I don't have to worry about it. I don't have to think about it. I don't have to tell my partner about it. I don't have to show any signs that I've taken a drug. So for us, that is some of the important things about this twice yearly injection. And we're looking forward to it.
So also about ease of use. What was the protocol around routine HIV testing? How did you manage that? And how do you see that playing out in the real world?
That's an excellent question. I do want to just say one fact, which is over 90%, so like 92% of people adhere to their injection visit. But the oral adherence was approximately 30%. And that tells you exactly what Yvette said. Women came back for their injections. They were not taking their pills, probably because they didn't want somebody to see the pill bottle. It was hard to remember every day with all the other competing priorities like school and work and all the other things going on in young women's lives. So, and stigma and all these other reasons, not wanting people to hear the pills jangling on the bus, right? So it is set it and forget it. You go discreetly to the clinical site, you get your injection, you leave, no one has to know. So I think that was responsible, exactly what Yvette was talking about, for the differential adherence to study products, right? People did a better job of receiving the injection than being able to take their pills. Now, some people did a good job of taking their pills, but the majority overall did not.
And we know that that's the case right across the world with oral PrEP, isn't it?
Yeah.
It's
It only-
episodes of risk type thing.
Or it only works if you take it.
It only works if you take it,
Right.
yeah.
But back to your question about HIV testing. So this is really important, especially as we think about where to make, we want lenacapavir, if it's shown to be effective in Purpose 2, to be approved for use in the places where HIV incidence is the most, where people are most disproportionately affected. So in order to do this study, we had this screening incidence cohort. There you got regular HIV testing. If you were negative, you were evaluated with other medical eligibility criteria for can you get the study products, and then randomized into the clinical trial. If the person acquired HIV or was shown to be HIV positive, they were immediately referred to clinical care. But for the people who were positive, their samples, we tested with a special assay called a limited antigen avidity assay, or recency assay, and that allowed us to calculate the background incidence, which allowed us to evaluate. Okay, so in that HIV testing process, that was to get into the study. And then you had a baseline visit, which was 30 days after the screening cohort, an average of 30 days, or that was the limit of 30 days. Everybody got 4th generation antigen antibody testing provided by us, rapid point of care. Then they got a blood sample sent to the central laboratory, and they had an instrumented laboratory 4th generation antigen antibody test. Then they had, if that was positive, they had an HIV 1 to 3rd gen antibody serology differentiation assay that tells the difference between HIV 1 and 2 and acts like the Western blot did in the old days to confirm the HIV infection. If there was discrepancy between those two tests, they got a qualitative HIV RNA, because only qualitative RNAs are approved by the US for evaluating or for diagnosing HIV infection. They also got a quantitative RNA, and we stored samples at every visit. So if somebody had an HIV test or set of tests that indicated they acquired HIV, we went back and we tested their retrospective samples to see when did that viral load first turn positive, because as we all know, there's an eclipse period, there's a window period where the virus is positive before the antigen antibody tests show positive. So that allowed us to really confirm not only that the person had HIV, but their closest date of diagnosis or date of positivity on the RNA tests. And then throughout the study, we did the testing the same way, which is the CDC approved algorithm for testing. So 4th gen rapid, 4th gen at the lab, HIV 1-2 differentiation antibody assay, and then qualitative RNA if there's discrepancy. We did not do every time quantitative RNA testing, but we saved samples so we could go do it backwards in the people who got it. And we did that very intentionally because we didn't think it was necessary based on our experience with Discover. And the Discover trial, a paper just came out written by David Wall and Christoph Spinner, the co-chair, with our 144-week data in cisgender gay men and transgender women of FTAF and FTDF that also shows that adding quantitative viral load does not improve the efficiency of diagnosis. So based on our experience in the PrEP field and our experience with our own data, that's the way we set up the study.
I was laughing because in a separate life, I've been heavily involved in this question of do you, in the age of PrEP, do you bother with HIV tests, or do you go to rapid viral load tests? And I've still got the bite marks from that. But in real terms, so a woman in Limpopo would get her injection twice a year. How often would she I mean, are we at the point where we can actually recommend this or say this? How often in between that would she be monitored?
It's a great question, and we have to dive more into our data. Obviously, when one does a clinical trial, you have the most rigorous safety lab and HIV testing because we're doing a trial for regulatory approval. In terms of clinical practice, I think to answer your question, we'd have to look at, did anyone get infected, acquire HIV in that period in between injections? Because if they didn't, why do you have to test them for HIV if they have Len levels that are protecting them? Now, our data can't answer that question because there were 0 infections.
Right.
However, as Linda Gale noted from the stage, after the DMC told us that we met the interim efficacy criteria and we could start offering people open label lenacapavir, we started to, and over 860 women have already chosen to go on to open label lenacapavir, and they're going to be followed for an extensive period of time. So we'll be able to see more real world, although they'll still get the rigorous testing, but in open label, real world kind of clinical setting, what happens with lenacapavir and what happens with HIV infection. And I think if we continue this kind of trend, then we could de-link the quarterly testing recommendation when you have a product that purportedly protects you for 6 months.
Yeah. So all of this then gets us on to the $36 million question, doesn't it? Access and availability, because I suppose is there ever a downside when you have such iconic data being presented? Well, you certainly create demand, don't you?
Yeah.
And as you've pointed out, the compound is still in extensive R&D. So, you know, the calls on making it available now, right now, are obviously not ones that can be rapidly responded to. But as Yvette said at the start, you've given a lot of thought to what the access strategy should be. What's your thinking there? What's the company's approach going to be?
So in terms of, before we talk about access, we have to talk about regulatory approval. And before we talk about regulatory approval, we have to talk about the package of evidence required for regulatory approval. And that includes everything we have so far with Purpose 1, but it also requires Purpose 2. So we usually need 2 pivotal phase 3 trials for regulatory approvals. And Purpose 1 was in cisgender adolescent girls and young women aged 16 to 25. And then we have Purpose 2. Purpose 2 is in folks 16 and older, cisgender gay men and other men who have sex with men, transgender women, transgender men, and gender non-binary people, all of whom have sex with male partners at birth, people who are assigned male at birth. Sorry. And that's taking place in 7 countries. And we need the results from that before we would file regulatorily. Now, assuming, knocking on wood, that Purpose 2 reads out in a way that indicates lenacapavir efficacy in those gender diverse individuals, then we would file for regulatory approval. And we're working really fast because I know personally how important this product is, and I know it works in women, and we're doing whatever we can to accelerate our process. I mean, we just learned on June 20th that, and we're already here, the data has been analyzed, published in the New England Journal today, which is very exciting, concomitantly with Linda Gale's presentation. And I think that just indicates the speed at which we're working, because we feel like we have a responsibility to do this as quickly as possible. So next steps. If they both read out positively, we're going to file with the regulatory agencies. We're going to use a process that we're negotiating, obviously, and have to do all the steps for, where we work with not only the FDA and the EU, but the EU Medicines for All pathway and WHO pre-qualification. And that will allow us to get it approved for a group of countries. And once it gets on the WHO pre-qual list, it's a lot easier. At the same time, Gilead has committed to, and this is all in our access statement available on our website in much more detail than I can describe it, but Gilead commits to providing lenacapavir to where it needs to go while we are setting up the current relationships with all our direct voluntary licensees, basically our generic companies. Now, we've worked with generics for years, right? For remdesivir, for hepatitis C, for HIV treatments, and for all the other products that Gilead makes that we have gotten worldwide, including very quickly in a pandemic. So we have a robust network already of our generics, and we want to work with them directly because our focus really is speed, volume because of the demand that's being hopefully created, and high quality lenacapavir that gets to the people who need it the most. And the countries and the people in the study are prioritized in the first wave because they contributed to the helping get at the data.
Right. Now, one of the questions that's come up this week is, yes, Gilead has a network of generic partners, and it's something that has been built over the years. It truly is impressive. I don't think anyone can dispute that. The company's taken a slightly more distant view of working with something like the medicines patent pool. And again, not to put you on the spot necessarily about that, but what was the reasoning behind that, or are all doors open?
So I was not involved in the direct decision-making process, but what I know is we want to get this product to people as quickly as possible. And all the people in my company who are experts in manufacturing and supply chain and all the stuff that is not my area of expertise, they think that the proposal that we have is the best way to get it to people as quickly as possible. We have worked with MPP on multiple other things. In fact, we helped support MPP getting started back in the day. And I think the reason MPP was started was to help companies that don't have their own direct relationships with voluntary licensees. And I think MPP is a great mechanism for companies who don't have the expertise and the relationships that and we've also worked with them when it makes sense. So, and it doesn't mean that we won't work with them in the future. In this particular instance, our goal really is volume, speed, because of how important we think this product is.
Yeah, absolutely. Yvette, your reflections on the company's strategy. I've no doubt you will be holding both them and us accountable. What are you going to be looking out for?
I think also, Ben, this GCAG and Gilead comes a long way. So every step of the way have we engaged with them. And it took a long time for us to talk access and to talk, licensing for generics. And we took, I think, over 5-6 months just discussing what is going to be the best way forward. And I would like to also challenge Gilead in a sense to say, all right, you want to do it this way, speed, access, and how it becomes available. How quickly does it become available? But think around, what would it mean? Are you opposed to working with both Moupali?
No, personally, I'm not opposed. I'm a Big Ten kind of person. But what I thank you for reminding, the people who made this initial decision did tons of outreach, including with the GCAG, and Yvette was involved in many of those discussions, talking with the people who are the experts, right? You know, they're a different set of people than us who are brilliant at what they do. And they took advice, and they also talked to a bunch of the big global multinational entities and partners and funders. So this was not a snap decision, and it's also a work in progress. This is what we're going to do now. Doesn't mean that we can't do something differently. And we really want to also have broad, sustainable access. So I like to think about the next hoop because that's how I trained. But we need to think about the hoops into the future. So we're not opposed. We want to do what is the right thing for the people who need it the most.
Yes, and for us, importantly, is it needs to become available. We know there are processes, like Moupali say, it still has to go. We still have to wait for purpose 2. We still have to go through FDA, and after that, all our regulatory authorities. But importantly is that we need to engage and we need to start talking with those important people on the table. In 2019, 5 years ago, when this started, we spoke already around price. We already spoke around access. So we are working together and we hold Gilead accountable and any producer of HIV prevention methods. That's what the community has to do, and how we hold each other accountable.
And we're a long way off from talking price, but it is interesting to know, if I've got this right, Moupali, that Lenacapavir is easier to make than some of the other long actings that are out there.
This is not an area of my expertise, but I do know it took us from 2006 to figure out what lenacapavir was, right? Screening thousands and thousands of molecules to get to making lenacapavir. And when we got the product that eventually became lenacapavir, this was in the early 2009, 2010. It was very insoluble, which was not great back then, because back then we were in the STR era. We were trying to take 20, 30 pills a day, turn it into one pill once a day. So having something insoluble was not great. It took a long time to figure out how to make lenacapavir, which is insoluble and highly potent, into an orally bioavailable pill. And I know how hard that was, because it took from 2010 to 2016 where we had that candidate. So, and this is by our process chemists and the people who are expert in making those pills. And I think that the formulation of lenacapavir, there's lots of excellent papers and articles about it. I don't know enough about the other long-acting drug to understand whether our process and their process is different. But I do know that remdesivir, which is an injectable that we make, is something that we were capably able to license to generics, and they were able to make it available so rapidly during the pandemic. So I know that it's possible to make injectables rapidly available through our network.
Well, I guess we've reached the top of the hour. I know you both have other commitments to get to.
Yeah.
Moupali, what a real privilege and honor it has been to have you on the show. You are one of those people who is able to communicate complex scientific concepts in ways that even I can understand. So I am so grateful.
Yes.
And of course, I'm always grateful to you, Yvette.
Yes, but also can you acknowledge the black and brownness on this panel, please?
Yes, absolutely.
Yes, and it's about time that the research and the scientists look like the people that we are researching for.
Couldn't agree more. I mean, actually, in a way, if you think about it, so gay man, you are Bengali by origin?
Yes, Bengali, Indian American. Born and raised in New York, now live in San Francisco, but my family is from India and Bangladesh.
Thrilled to hear it. Well, the Californian component, certainly. That means we're neighbors. And of course...
Yvette from South Africa, Limpopo, a village girl. And here I am fighting for my community, fighting for women, and fighting for the future of my own children. And I just to mention, my daughter is here, and I hold her accountable for ensuring my daughter does not get HIV, because I got onto PMTC to prevent that young person to get HIV. So yeah, and she's doing other things. She's not walking around the conference saying, "Hi, my name is Waisha and I'm living with HIV," and I'm proud. I want it to stay that way.
Yeah, amen to that.
Thank you for the honor of being able to come talk to you guys about these exciting results.
It's been a real pleasure. Thank you. Well, that's it for this episode. Thank you to Dr. Moupali Das from Gilead, and of course our very own Yvette Raphael. Thanks to Waisha Raphael, who of course is our production coordinator, to Will Lansdale, who is our production assistant, and of course the man who makes the magic happen, Eric Espera of Newsdoc Media, our producer and director. So with that, have a great day and a safe day, everybody.